Hello, my name is Jennifer Alonso, and I am a student of Lisa Friend's Microbiology Lab online course this semester, and this video will be a discussion on the Gram stain. The major difference between gram-positive bacteria and gram-negative bacteria is the physical properties of their cell wall. The peptidoglycan layer of gram-positive bacteria is thick, while the peptidoglycan layer of gram-negative bacteria is thin and surrounded by an outer membrane that contains lipopolysaccharides. The Gram stain is used clinically to divide bacteria into two subgroups, which will allow identification of a bacteria, an infection, and then will assist with the treatment, the course of treatment, and antibiotic therapy to combat that infection. The Gram stain is as follows. Step 1 is to prepare the bacterial smear on a slide. Step 2 is to apply the first dye to the slide. The first dye that is used is the crystal violet dye. This dye will stain all of the cells purple. This dye will be applied to the slide and be allowed to sit for 1 minute before it is rinsed with water to remove any excess dye. The third step is to apply the iodine, which acts as a mordant. The iodine binds to the crystal violet dye and forms an insoluble crystal violet iodine complex. This, the iodine is allowed to sit on the slide for 1 minute and then will be rinsed with water to remove any of the excess iodine. Step 4 is to apply the decolorizer. This solution is 95% alcohol. This solution will be applied to the slide for 2 to 3 seconds and then immediately rinsed with water to remove any of the excess alcohol. The final step is to apply the next dye called safranin. This will be applied to the slide, allowed to sit for 1 minute. And what this dye does is removes the outer membrane of the gram-negative bacteria, and then the cell will pick up the pink dye, and the gram-positive bacteria will retain their purple dye during this step. So at that time, the Gram stain is complete. You would blot the slide dry, and you will be able to view it and interpret the results via a microscope using oil immersion therapy or oil immersion microscopy. To identify the different subgroups of the bacteria, gram-positive and gram-negative. As I stated before, this is used to subgroup the bacteria into the two subgroups to allow for better identification of the bacteria, the infection, and how it will be treated. Thank you.